Doctor,
Ah,
the study of chromosomes and its abnormality.
okay, yeah. Study of chromosome. Chromosome apa? Kita tengok chromosome punya structure. Okay. Tengok abnormality dia macam mana. The chromosome number is all about cytogenetics. Okay. Okay, so it's a field of genetics that involve in microscopic examinations of the chromosome and chromosome rearrangement. Okay. So typically is they examine the chromosome compositions and also the particular in the cell and also organism. So this allowed the detection of individual with abnormal chromosome number or structure. What happened is why this is so important. Yeah, can you imagine if you have extra chromosome? What will happen? Okay.
Mutation.
Okay. What happened?
Genetic disorder.
Yes,
They
genetic
have
disorder.
abnormal mutation.
Contohnya?
Down syndrome.
Down syndrome.
Down syndrome. Apa lagi?
Turner. Turner
Syndrome Turner, Klinefelter's, right? Banyak kan? It's also used to distinguish between species. Kita dengan burung takkan sama chromosome kan? Yeah. So this is also one of the criteria that you can be used to study to distinguish different species. Okay, but not all chromosome rearrangements are bad. I need to tell you this. Like in plant, you know, some of the plant they have extra chromosome and surprisingly they produce better yield. Ada macam itu. Okay, but nanti kita I'm not going to go into that lah. Just to tell you that. Okay, so not all semua akan menjadi disorder ataupun masalah, not necessary. Okay. Now, so it's a fundamental to understanding the evolutionary history of a species. And although the contohnya gorilla and humans are morphology very different, but at the level of chromosome, the DNA sequence, they are very extremely similar. Kalau kita tengok di sini, okay? Kalau kita tengok the chromosome of humans, chimpanzee, gorilla, and orangutan, lebih sangat similar, betul tak? But morphologically, we are very different. Yes? Do you look like a chimpanzee or gorilla or orangutan? Well, not right. But you see, the chromosome structure are very similar. So this is important for if you want to study, why we study chromosome variations is there are for a few reasons. Okay, for example, they can have major effect on the phenotypes of an organism, like what we see here. Very similar chromosome arrangements, but result in very different phenotypes. Okay, and then they can have major effect on the phenotype of the offspring of an organism juga. Okay, contohnya Down syndrome. Orang mak bapak normal tapi hasilkan Down syndrome, the phenotypes also very different to the parents kan, and also have been an important force in the evolution of species. Dia merupakan satu daya yang penting, okay, dalam evolutions of species, okay, penghasilan species baru, okay. So where does this, the words come from? Is chromo is basically is a color in response to dye, and then som is the body. Okay, so chromosome in eukaryotes have been the traditional subject for cytogenetic analysis because they are large enough to be examined with light microscope. This light microscope actually kita boleh tengok lah, tapi this one is as electron microscope punya level. Okay, you can actually see, but this one is electron microscope lah. Okay, you can see. Macam you cakwe, betul tak? Macam cakwe kan? Okay, cakwe tapi on top ada you banyak cheese. Macam kita punya goreng pisang yang banyak cheese atas tu. Okay, and chromosome of different species differ in the number and information contents, of course lah. That you can see the prokaryote. Okay, in prokaryote, the single chromosome with plasmid, and it's a circular matter of only of DNA and found in cytoplasm and copy its chromosome and divides immediately afterward. But in eukaryote, they have many chromosomes. And usually they can be linear and made of chromatin, and then a nucleoprotein where the DNA coil around the histone protein. Later we'll discuss that. Okay, and found in nucleus. Right, you can see the different prokaryote they found in the cytoplasm, but eukaryote they found in the nucleus, and then the copies chromosome. Then the cell will grow and then goes through the mitosis organize the chromosome into equal groups. Okay. It's just introduction. How about viral chromosome? Is basically a virus is a nucleic acid surrounded by a protein coat. So nucleic acid may be double-stranded, okay, or single-stranded, and it may be linear, okay, or circular in a single molecule. So basically, cerebral importance of viral chromosome is that the T event fetch is that the T events the event fetch kita ada T2, T4, and T6, and they have very similar structure and have double-stranded DNA genome composed of one linear DNA molecule surrounded by a protein coat. How about for this 174 fetch? It's very small, and there's a simple virus with a short single-stranded DNA chromosome, and it has a base composition that does not fit the complementary base pair rules. Okay, so viral pun ada chromosome dia sendiri. Okay. But the structures is very different compared to the eukaryote lah. Right? So prokaryotic chromosome, the genomes is one circular double-stranded chromosome, but some are more exotic, with the main chromosome and one or smaller one. Okay, and when a minor chromosome is dispensable to the life cell, then it is called the plasmid. So contohnya dalam bacterial, dia ada plasmid lah. Okay, dia ada plasmid sendiri yang circular punya. Okay, for example, the Borrelia burgdorferi, which is a Lyme disease in humans, it has a 0.91 megabases linear chromosome plus additional 0.52 megabases of DNA in 17 different linear and circular molecule. And for Agrobacterium tumefaciens, which result in the crown ground agar disease of plants, they have 3.0 megabases of circular chromosome and 2.1 megabases of linear. So you see some of these bacterial, okay, or fungus, they have circular one and also the linear one. Okay, they can have both actually. So prokaryote chromosomes are highly supercoiled to help it to fit into the cell because the cell is so small. So they need to be extremely supercoiled so that it can fit right into the cell itself. Okay, contohnya bacteria. Bacteria very small, right? So if they have the chromosome cannot be too large. If they're too large, they take out so much of the space. And that's why they need to compact in a very supercoiled form so that it can fit right into the cell. Okay, so far okay tak untuk ini?
Okay, Doctor.
Okay, Doctor.
Okay.
So, supercoil tahu tak? Supercoil. Inilah supercoil. They coil together and then they're very, very packed. Okay, they packed together. Nanti kita akan tengok bagaimana the chromosome rearrangement. So eukaryotes, as I said, they have multiple linear chromosome located in the nucleus. But in prokaryote, it's located in the cytoplasm. Very good. So the number of chromosomes is characteristic of the species. So most have two versions of each chromosome, and this is why we call diploid. Okay, 2N. Okay, all humans will have 2N kan? Okay, only the gamete have 1N. Is that right?
Yes.
Okay, so diploid cells are produced by a haploid gamete that fuse to form a zygote. So when they form the zygote, they call the genome restoration. So then undergo developments, forming a new individual. So for example, a diploid organism, humans, for example, the 3 pairs, and chromosome melanogaster, there are 4 pairs. Okay, and use a Saccharomyces cerevisiae here, they have a haploid of 16 chromosomes. So you see, all species, they have different chromosomes. And the pairs in diploid organism are homologous. Okay, the homolog chromosome, and one member of each pair homologs is inherited from each parent. Contohnya, you get half of the genome from your father. So you also have one end from your father, one end from the mother, and become the 2NU. Right? So you have the diploid genome. Okay, so chromosomes have different genes and do not pair no homologous chromosome. Okay, and they differ in size, of course. So each chromosome, dia ada chromosome yang lebih kecil, ada yang chromosome yang lebih besar. So each has a contractions called the centromere that is used in segregations during mitosis or mitosis. Okay, masih ingat tak kita ada centromere? Yeah, centromere.
Yes,
And then the centromere, the location can be very different. Okay, so locations are useful for identifying the chromosome, because some of the chromosome, they are metacentric. Metacentric means the chromosome, the centromere is approximately in the center of the chromosome, producing N2 equal arm. Contohnya di sini. So ini lebih kurang 2 panjang kan? The P arm and the Q arm kan? The length is almost even. Okay, lebih kurang sama panjang dia. This is what you call metacentric. For submetacentric, which one arm is somewhat longer than the other one. Contoh di sini, this is the P arm, this is the Q arm. So the P arm here is slightly shorter than the Q arm. Can you see my pointer?
Yes.
Yes,
Okay.
doctor.
Okay.
Yes.
Okay. Another one is the acrocentric. Acrocentric is like the apa tu, have one long arm and a very short stalk. Often it's like a bulb, like a satellite. Contoh di sini, dia macam satu bulb saja. Okay, this is what you call acrocentric. Okay, another one is the telocentric, whereby have only one arm because the telomere is at the very end. Contohnya di sini. Okay, so these types of rearrangement can help us to identify the number of the kedudukan of the chromosome. We can name chromosome. Setiap chromosome, ingat ya, dia mempunyai feature yang berbeza. Okay, so this is how we look at the structure of the chromosome. Okay, ada maksud dia sendiri kenapa dia macam itu. Kenapa dia metacentric, kenapa dia submetacentric, kenapa dia acrocentric dan juga telocentric. Ada sebab dia. Okay, so can be identified by the size, the shape, the positions of the centromere, and also the bending pattern produced by a specific trend. Now here we use GIMSA, okay, what you call G-band. Okay, of course we have more than that. Okay, kita to look at the characteristic of the chromosome. Not only the G-band, we can have other methods to identify them also. Okay, but I'm not going to talk in detail lah. Okay, so chromosomes are analyzed by organizing them into karyotype. So karyotype is the one that we look at the number of the chromosome and also the appearance of the chromosome. What you call karyotype or karyotyping is an analysis. Okay, analysis. Chromosome analysis, we call karyotyping lah. If kalau orang kata, I want to do karyotyping, that means they are not what? Chromosome analysis. Okay, so for detailed identification, chromosomes are then always treated with the stain to produce the characteristic bending pattern. Okay, so example, the G bending. Chromosomes are exposed to the GIMSA. GIMSA is a type of dye lah. Okay, so it is a very specific for the phosphate group of the DNA. So some regions bend the dye heavily because of the dark band. Dark band, that means they're very condensed. And then some regions, they do not band, they do not die to the stand well. So they produce the light bending. Contoh di sini. Can you see here? You have the very dark bending, some very light bending. Nampak tak di sini? Can you see that?
Yes, doctor.
Okay.
Yeah.
This also contains a few chromosomes. So from there, you can see each of these chromosomes, they have different types of bending pattern, betul tak?
Yes, doctor.
Yes, doctor.
Okay. And then some in humans, you have about 300 G bands are seen in metaphase, whereby you have 2000 G bands in prophase. Why? So from there, you can see the stage of the cell. So each test of the cell, they produce different types of bending. And from the bending, you can tell which stage of the cell belong to. Yeah? Contohnya, mitosis, they have 4 stages kan? Masih ingat tak? Kita ada prophase. Ada lagi?
Anaphase, anaphase, metaphase.
Okay, kita ada, we have prophase, metaphase, telophase dengan
Anaphase.
Okay, four phases. Okay, and kita jangan lupa kita pun ada satu phase. Memang kita ada satu
Cytokinesis.
Okay, kita ada satu lagi di mana sel tu dia berehat. Okay, so other technique to visualize chromosome include R bending. R bending is the reverse of the G bending. You do the reverse. So the dark regions are euchromatic, whereby it is the guanine, cytosine, a GC-rich region, and the bright regions are the heterochromatics, which is a thymine adenine rich. And usually the chromatics, they are inactive, not inactive, and whereby the heterochromatics, they are actually more active actually. So kita ada C bending, whereby the genes are bind to the constitutive heterochromatins, so it stands the centromere. So kita stand centromere saja. That will provide the C bending. Okay, and also we have the Q bending, whereby it's a fluorescent pattern obtained from using a quinacine. Okay, quinacine for staining the pattern of the bands, very similar to that C in the G bending. The other one is the T bending, also used to visualize the telomeres. Another one is a silver staining, whereby silver nitrates stain is used for the nucleolar organization regions associated with the protein. So this yield a dark region where the silver is then deposited. So there are many, not only the G band can be used, but the R bending, C bending, Q bending, T bending, and silver staining can be used to analyze the chromosome characteristic. Okay? Boleh tak? So far okay?
Okay, doctor.
Okay, very good. So if you look at here, okay, so the karyotype of male and female are not the same. So female, they have 2 large X chromosome, whereby male, they have 1 large X and 1 small Y. Yes? Okay, so the sex chromosome are placed at the end of the karyotype, like here. Okay, if you look at this pattern, yeah, this is the male, this is the female. You can see each of these chromosome, the size different, isn't it? Like chromosome number 1 is much bigger than chromosome 16, 17, 18, 19, 20, 21, 22. Betul tak?
Yes.
Yes.
Okay, and then if you look at the X chromosome, okay, the X chromosome here and here, compared to the Y is much smaller than the X. Betul?
Yeah.
What does it mean? Kenapa ada kromosom yang lebih besar? Kenapa ada kromosom yang lebih kecil?
The position of the centromere.
No, it doesn't matter actually, the positions. Because kepanjangan dia, it doesn't matter. If they are a long chromosome, it doesn't matter kan. Sama ada dia metacentric, apocentric, it doesn't matter. Yes? It's not because of the positions of the centromere. Actually, if the larger the chromosome, the more gene is contained.
Oh, okay.
Because you need to fit in more gene kan. Logic or not?
Yeah.
So that's why you see X dengan Y. Of course, X lebih banyak gene dalam tu kan. That's why kita selalu kata X-link, X-link, X-link. Pernah dengar tak X-link disease?
Yes.
Okay, tak apa. Kau tak pernah dengar, tak apa, because the coming lecture nanti kita akan belajar tentang X-link punya disease nanti. Okay? So kenapa certain disease tu dia daripada mak? Ya? Certain disease dia daripada bapa. Dia ada X-link punya disease dalam tu. Kenapa ada certain tu kalau daripada mak, anak laki dia okey. Ya? Ada sebab, nanti kita akan belajar ratio dia lah. Okay? Okay. Now, kalau kita tengok kan the nomenclature, okay, bagaimana kita nak baca kromosom ni. Contohnya kita ambil example of chromosome 3. So, this one, yang ini, we will call P, petite, P-arm. We call it P-arm and Q-arm. Q-arm is Q lah, Q-U-E-U-E. We call it Q-arm. And then tengah-tengah ini kita panggil dia centromere. Okay? Di atas ni yang paling ujung ini is what we call P telomere and Q telomere pada kutub-kutub tu. Kalau dia pada kutub P, kita panggil dia P telomere. Kalau dia pada kutub Q, kita panggil dia Q telomere. Okay? Kita nak tahu kenapa kita bagi nombor-nombor macam ni ialah untuk kita senang nak tahu di mana posisi gene. Kita nak tengok lokasi gene itu. Contohnya kita, the regions of chromosome is called 3p22. Okay? Kenapa dia panggil 3? 3 maksudnya kromosom 3. Okay? And then, kenapa dia P? Kerana dia terletak pada P-arm. Yeah? And 2, kenapa dia panggil 2? Kerana dia pada region 2. Dia region 1, 2, 3 kan? Betul? So, dia pada region ini kan? Boleh faham?
Yes,
Okay?
doktor.
Dan pada region 2 ini, dia bahagi lagi kepada 2 region. Dan dia berada di sini. That's why ini panggil dia 3p22. 2 here. And with this region is 3p21. Kerana kita nak di sini. Kalau gene tu terletak di sini. Boleh faham?
Yes, doktor.
Okay. Dan kalau kita nak spesifik lagi, this region, kalau kita nak region ini, bateri hilang. Tak, saya tukar bateri dulu. Tak ada bateri. Tak. Nampak cerita kan? Lebih jelas lagi kan? Saya punya pointer ni, okey tak? Lebih jelas lagi kan?
Yes,
Yes.
doktor.
Okay, so kalau kita tengok ini, kalau kita nak region ini, kita panggil dia 3p22.1. Yes? Kalau ini, kalau di sini,
0.2.
ya, 3p22.2. And this is how we locate our gene. Kita kata, okay, kalau kita nak belajar satu jin, jin nama dia jin alpha. Jin alpha located at Q21.3 contohnya. Ataupun 21. Gantung lah. So di sana kita tahu, oh, I'm looking for chromosome 8. Dekat mana? P-arm atau Q-arm? Q-arm. Region mana? Region 22, 21. Point berapa? That's how we locate the gene. Kita nak tahu kedudukan jin supaya kita lebih senang tahu. Make sense or not? Okay? Boleh faham, Okay. Dan also, if we want to study the function of the gene, we always look at the neighboring gene. Okay? There must be a reason why these genes they reside together. Betul? Make sense or not? Gene they cannot function by itself, betul tak? Sometimes dia boleh control other gene or the gene being controlled. So usually we look at the surrounding punya gene to get a bigger picture of the function of the gene. Make sense tak? Alin, make sense tak?
Yes.
Yeah? Make sense kan?
Yeah.
So, bila kamu belajar fungsi jin ini, kita akan tengok kedudukan dulu. Lepas tu kita tengok siapa jiran jin ini. Okay? Because senang dia nak berkomunikasi. Jin pun berkomunikasi. Okay? Kalau dia jiran kan, senang dia berkomunikasi, betul tak?
Betul.
Tapi kalau dia jauh, dia susah nak berkomunikasi because jin ni tidak ada telefon. Nah, dia bukan macam kita call, hello, how are you? Bukan macam tu. Contohnya, kalau a gene reside in chromosome 1, susah dia nak communicate dengan chromosome 22, betul tak? Because they are too far away. Because they are reside on different chromosome. Kalau dia reside pada contohnya chromosome 3p22.1 dengan 3p22.2, dia jiran saja kan? So it's a big chance for us to understand whether these two genes ada any relationship ke tak. We can understand. Contohnya kita silentkan gene pada 3p22.1. We want to see will it affect the gene at 3p22.2 ke tak. Make sense? So, dari sanalah kita belajar. Sama ada jin ni dia ada fungsi dengan dia punya jiran ke tak. Adakah dia mempunyai hubungan? Okay, ataupun communicate. Adakah communication tu breakdown because we silent the gene atau we knock out the gene. Make sense?
Yeah.
Yeah. So, kita kena sentiasa fikirkan. Okay, kalau kamu betul-betul minat dalam bidang genetics ni, you need to think ahead lah. Kalau tak minat tak apa. I cannot force you lah. Because I cannot force anyone. Because everyone liking is different. I cannot force you to understand. But I force myself to make you understand. Because that's my responsibility. Itu saja. But if you resist not to understand, I cannot break your head, open your brain and then cuci dia. I cannot force you, but I'll try my best to make you understand. Okay, so nomenclature okay Boleh sekarang?
Yes.
You can name the gene sudah kan?
Yeah.
Okay, very good. So we go next. Okay, so in eukaryotic chromosome, the form of chromatic chromosome changes through the cell cycle. Contohnya kita ada G1 phase kan? So each chromosome is a single structure. Okay, masih di sini. After the cytokinesis kan kita ada G1 and and G2 kan. Lepas tu kita ada mitosis dekat sini. So setiap di sini kromosom punya condition berbeza. Sama dia di G1, S1, G2 dan mitosis. Betul tak? So the pattern of the chromosome also different at each of this stage, the cell cycle. Are you agree with me?
Yes.
Yeah, agree kan? So chromosome duplicate into sister chromatid but remain joined at centromere through G2 di sini. Kenapa? Kerana dia double sudah. Kenapa dia double? Kerana dia nak sediakan diri dia untuk proses mitosis. Dia double up, betul tak? Ya, untuk persediaan pada mitosis. Kerana dia nak hasilkan sel-sel yang baru. Kalau dia tidak double up nanti, macam mana dia nak pisah nanti menjadikan 2N nanti kan? Ya, so at M phase, okay, so sister chromatid was separated into daughter chromosome. Contohnya di mitosis ni. Ya, dia kena separate. Kerana dia nak hasilkan sel yang baru. Kalau tidak, dia tidak double up dekat sini. Macam mana dia nak separate nanti? Nanti satu tu dia kosong, tak ada kromosom. Satu tu penuh dengan kromosom. Dia tidak boleh bahagi nanti. Yeah. So in G1, the chromosome are linear double-stranded and contain about twice as much as the protein as the DNA by weight. And then the DNA protein complex is called chromatin. Okay, and it's highly conserved in all eukaryote. Right? So now come to the chromatin structure. It is slightly complicated sikit. Okay, nanti saya akan explain lah. So chromatin formations involve small protein called histone protein. Pernah dengar histone protein? Okay, that assists to condense the DNA so it will fit into the cell. Okay, protein ni dia sangat penting kerana dia yang akan membantu untuk compact kita punya DNA. DNA kita merupakan macam satu benang yang panjang, betul tak?
Yes.
Ya. Nah, kalau kita tidak compact ya, nanti dia berserabut nanti dalam sel kita. Dalam nukleus kita, dia berserabut. So, dia mesti compact, se-compact yang mungkin supaya dia boleh fit into the nucleus. Because nucleus kita kecil lagi daripada sel kan. So, histone are abundant, dia memang banyak. Okay, and small protein with a net positive charge. DNA kita positif kan negative charge?
Negative.
Tidak tahu.
Negative. Negatively charged.
Negative.
Positif kan negatif?
Negative.
Cuba type, saya nak tengok siapa kata positif negatif. Jangan ikut kawan. DNA kita negatif ke positif? Cuba type dalam chat box. Siapa tak type tu saya rasa dia tidak dengar lah. Ataupun dia, wah semua bagi negatif kah? Betul ke ni? Ikut kawan ke? Okay. DNA kita negatif kan? Okay. So, you tengok histone tu positif. Positif dengan negatif dia akan attract. Kalau dia attract, maka makin padatlah dia. Kerana dia tarik di antara satu sama lain kan. So, makin padat. Betul tak?
Yes.
Agree kan?
Yeah.
Kalau tidak, dia akan loose. Dia akan rongga. Kalau dia tidak tarik antara satu sama lain. So, you see, so smart. Dia gunakan charge itu untuk menjadikan dia sangat-sangat padat. Okay, to condense them. So kita ada 5 main types of protein histone. Kita ada A1, H2A, H2B, H3 dengan H4. Okay, alright. So by weight, the chromosome have equal amount of DNA and histones, and they are highly conserved between species. So histone organize the DNA, and condensing it and preparing it for further condensation by non-histone protein. So this compaction is necessary to fit the large amount of DNA, which is 2 meter, which is 2.5 feet in humans, into the nucleus of a cell. Just imagine 2 meter, panjang tak the DNA? Macam mana dia boleh fit into kita punya nucleus tu? Can you imagine? So maksudnya dia mesti sangat padat. Agree?
Yeah.
Okay. So both histone and non-histone protein are involved in the physical structure of the chromosome. So non-histone protein is generally named for other positions associated with the DNA, and this is the big group with some structural protein and some that bind only transiently. Transiently itu maksud dia bind sementara saja, lepas tu dia akan keluar. Dia bantu saja dalam compaction tu. So non-histone protein very widely, okay, even in different cell from the same organism. So dia setiap non-histone protein ni pada setiap sel tu dia mungkin berbeza. Ya? Faham tak? Dia bukan semua sel sama non-histone protein. Dia berbeza. Okay? So most have a net negative charge and then bind to by attaching to histone protein. Contohnya kalau kita tengok di sini. So kita ada histone core molecule. Okay? By kita ada H2B, H2A, H4, H3. Okay? Ya, ini adalah core, histone core. Maksudnya dia ada dalam pusat dia. Okay? Dan ini kita punya DNA, kan? Yang benang ni, ini DNA. Nampak tak? The linker DNA. Yes?
Yes.
Okay? Dan tadi kita adalah satu lagi kan? Histone protein H1 kan?
Yeah.
Yes, doctor.
H1 tu dekat mana? Dekat sini. Yang warna kuning ni. Nampak? Dia tidak tergolong dalam histone core. Histone core terdiri daripada H4, 3, H2A, H2B. Tapi H1 ini dia tidak tergolong, ingatlah, tidak tergolong dalam histone core. Macam core subject lah. Dia bukan core. Bukan dalam utama. Kerana apa fungsi dia ialah dia adalah digunakan sebagai satu membantu dalam apa tu, sebagai satu sokongan, supporting, supaya dia, kerana DNA dengan histone core ini bila dia lilitkan, dia kena something to support dia supaya dia lagi kuat. Boleh? Itu one of the functions of H1, histone 1. Faham tak? Hello?
Yes,
Yes.
doctor.
Boleh ya?
Doctor.
Okay, so two molecule each of histone H2A, H2B ini to form nucleosome core. Okay, ini pun dikenali sebagai nucleosome core molecule. Ini. Okay, and the DNA wrap around H, which is 1/3/4 times for a 7-fold condensation factors. So nucleosome core are about 11 nanometers in diameter. Okay, so H1 further condenses the DNA by connecting the nucleosome to plug the chromatins with the diameter. So yang ini, so fungsi dia ialah dia condense lagi, dia mengetatkan dia lagi for additional 6-fold condensations. So the solenoid model proposed that the nucleosome form a spiral with the 6 nucleosome per turn. So dia dalam satu turn tu dia ada 6 nucleosome. Okay. So this is how they compact it. Kamu ada tengok video tak yang saya share? Bagaimana daripada DNA dia membentuk menjadi kromosom tu. Ada tak? Amazing kan? Bagaimana benda yang 2 meter panjang boleh dimuat ke dalam nukleus kita. Dan dalam badan kita, kita ada 46. Bayangkan. Ya? Amazing kan?
Yes, Doctor.
Ini kamu sendiri dekat rumah boleh buat satu model lah. Ambil satu tali ataupun thread benang. Lepas tu kamu ambil satu ball. Okay? Lepas tu kamu bahagikan ball tu kepada 4 bahagian. Kamu boleh warnakan dia. Lepas tu satu bahagian menjadi H3, satu H4, satu H2A, satu H2B. Kamu lilitkan dia. Lepas tu kamu ada satu lagi menjadi H1. Kamu boleh buat you punya solenoid punya model tu. Kalau physical, I'll teach you how to do it. But oleh kerana tidak dapat bersemuka, so actually you all sendiri boleh cuba buat lah dekat rumah. Actually paling senang kan, bagaimana kita buat, kalau you nak belajar ini. Gunakan benda yang paling murah. Do you know how to do it?
No, Doctor.
Kamu tahu tak gandum? Tepung gandum?
Yes.
No.
Tepung gandum boleh jadi dough tak?
Ya. Boleh.
Kamu pernah
Boleh.
pergi ke Toys R Us yang beli dough tu ke tak?
Pernah masa kecil.
See? Dough tu kamu boleh buat warna sendiri kan? Boleh letak pewarna kan?
Ya.
Dan you boleh buat you punya solenoid punya model lah. Satu tu you guling dia supaya dia semacam thread like panjang menjadi DNA. Lepas tu you satu tu you ambil menjadi 4 bahagian dengan 4 warna berbeza, menjadikan dia menjadi you punya histone core. Ya. Lepas tu you lilit lah pada you punya histone core. Lepas tu you buat lagi satu yang warna berbeza untuk menjadikan H1 tu. Then you can have your own solenoid lah model. Tak sampai RM1 you punya modal tu. Senang kan? Pewarna tu berapa sen saja. Ya?
Yes, Doctor.
But too bad. I cannot teach you because tak ada dalam physical. Kalau physical memang kita boleh buat macam tu. It's more interactive lah, lebih seronok lah. Karena kalau kamu gunakan tangan you and buat kan, actually you have different types of experience. You lebih faham lagi if you ada hands-on tu. Kalau if you don't have good imagination kan, you pun tak tahu apa yang saya cakap. Tapi dekat rumah boleh tak? Berapa sen saja tepung tu. Ya, dengan pewarna 4 jenis pewarna, mungkin 5 jenis pewarna, lepas tu kamu buat solenoid.
Doktor.
Actually ini boleh menjadikan satu assignment juga. Okay? So assignment tu ialah, tapi kamu jangan buat lah. Jadi kamu take foto. You punya, kalau saya nak buat lah, I would ask you to make solenoid punya model dengan menggunakan the dough that you make. Because very cheap. RM1 cukup. Tepung berapa saja, tak perlu sampai 1 kilo you pakai. Ya, mungkin 100 gram dah cukup. And then take photo. Cantik kan? Kalau you nak buat lah. Kalau you tak nak buat, tak apalah. Okay? But it's very interesting. Okay? To make you understand. Okay? I just give you idea lah kalau you nak buat benda macam ni. Okay? Because dekat rumah memang that's how I teach my children lah. So that they're hands-on, they do it, and then they have better understanding actually. Okay? Rather than I just talk and then I don't know whether you understand or not. That's why I keep asking you, do you understand? Agree ke tak? Okay? So, inilah contohnya solenoid punya model. Okay? Yang ini dalam bentuk electron microscope lah. Kalau kita tengok. Tapi ini model dia. Maybe if you are creative, you can make, apa tu solenoid model of bun. Now, bun. My nucleic acid, okay, my chromosome bun. You never know. It's very interesting tau kalau kamu pandai buat kuih ataupun pau. You boleh buat bentuk macam ni. Ya, a scientific punya, product nanti based on the model. Tapi usually kamu tidak pandai buat lah, saya tahu. Kamu tunggu mak buat saja. How many of you know how to bake?
I do.
Pandai
I know
baking
how to do that. Yeah.
Good.
During
Because
MCU.
cooking is fun.
Yes.
Cooking is
Tapi-
fun when it's not a duty.
Right? It's not a duty. Really. Let me share with you. I cooked for my family when I was 11 until now. Yeah. Okay, never mind. Teruskan. Okay, this is how DNA is packaged. Okay, daripada ini panjang, lepas tu dia kelilit dia punya histone, lepas tu come with H1, hold it and buat dia menjadi satu yang sangat padat, lepas tu menjadi macam ni. This is how DNA is packed and made into chromosome. Ya. Bayangkan daripada sini ke sini, okay, adalah 2 meter. And then pada hujungnya dia buat macam ni lah. Okay? Can you see the picture or not? How DNA is packed into a chromosome?
Yes, doctor.
Yeah.
Very interesting kan?
Yeah.
Can you imagine kalau dia tak mau macam kamu orang, dia malas, I don't want to pack. I don't want to be packed, dia kata. So what happened to your DNA? Kalau DNA kamu tu degil, I don't want to stay isolated. I don't want to make friends. I don't want to be packed. I don't want to go into jail, dia kata. Mampus lah you punya DNA itu. Yes? See?
Yeah.
All these processes are tightly regulated to make a perfect organism. Yeah, it's not easy, you know. That's why you must love biology. This is how we appreciate life. From all these processes, if you really appreciate it, you will appreciate your life. Okay? Okay. Move on. This is the chromatin structure. Okay? I'm not going to talk much about this because this is very simple. Yeah? Chromatin. Okay? Macam mana they pack right into this, and then the full condensed chromosome is about 10,004 shorter than 400 thicker than the DNA alone. Bagaimana dia boleh buat macam itu? Okay? The chromatin punya structure. Ini pun sama. In the summary. Okay? From here, DNA they pack, right? Beads to the string, and then they make into the chromatin fiber. Okay? And then at the end dia macam satu daripada DNA kepada chromatin. Apapun kromosom. Satu daripada kromosom, dia unleash dia, and then dia menjadi DNA macam ni. Ya? Like DNA replication. Dia kena bermula daripada kromosom ni. Dia mesti have something to unleash dia nanti. Right? To open it up. Okay? Because they are very, very tightly packed. So how are they going to open it up for the process of replications later? Yes? Just imagine now. In our body sekarang, right? Thousands and thousands of the similar processes is concurrently happening. But you don't feel it. Kalau chromosome ni dia gegel, I'm not going to let you open it up. I don't want to loosen it. So bagaimana protein synthesis boleh berlaku kan? Right? So the protein will not be produced. When the proteins is not being produced, macam mana? So we get sick lah. Right? Okay? Contohnya the protein that produce hormone insulin. So insulin at a very low level. So what will happen? You have high glucose in your blood. And insulin semakin rendah, at the end you get diabetes. Ya? Contohnya lah example. Okay? Yang ini euchromatin dengan heterochromatin. Okay? So euchromatins, okay, the chromosome cycle affect DNA packing with DNA condensing for mitosis and meiosis, and then decondensing during the interphase. So they're condensing during mitosis, meiosis, and decondensing during interphase. Okay, kenapa interphase dia kena ada? Dia kena double up. Okay, so chromosome are most condensed at the metaphase when the loop domains are vertical and the chromatic has a diameter of about 700 nanometers. And the non-histone proteins form the scaffold for this additional condensation. Okay, for this, upper to euchromatin, it condenses and decondenses with the life cycle. So it is actively transcribed. Okay, and like repetitive sequences, and euchromatin account for most genome active cell, whereby the heterochromatins remain condensed throughout the life cycle, and then is replicated later than euchromatin. So it's transcriptionally inactive. That's the difference between euchromatin and also heterochromatin. Satu lebih aktif, satu inactive. So heterochromatin ada dua activities, which is constitutive heterochromatin, and satu lagi facultative. Apa tu constitutive is that occur at the same site of both homologous chromosome of a pair, and it's mostly repetitive DNA. Whereby facultative, there varies between the cell type or developmental stages, or even between the homologous chromosome. So it contains a condensed, that's inactive euchromatin. So like, for example, bar body dalam sel kita. Okay? Dalam female punya sel lah. Okay? So, apa tu centromeric dengan telomeric DNA? Okay? Centromere and telomeres are the chromosomal regions with special function. Okay? So, centromere basically is the site where the spindle fiber attached during the mitosis and meiosis, and they are required for accurate segregations of the chromatids. Okay, because chromatids dia akan membahagi semasa mitosis dengan meiosis. So itu tempat di mana dia kena bahagi dengan sangat accurate. Okay, and telomere needed for the chromosomal replication and stability, because telomere basically is to confer the stability of the chromosome. If chromosome no longer stable, so dia senang putus. Sangat bahaya tu. So telomere are generally composed of the heterochromatins and interact with the both nuclear envelope and also each other. So simple telomeric sequences are short and species specific and tenderly repeated. For example, the tetrahemina, which is 5' is TTGGG3 for 3', and in human its 5' is TTAGGG. So it's very species specific. Okay, satu TTG, satu TTA. Dan belakangnya GGG dah sama. And Drosophila pula is unusual in having telomere composed of transposition. Okay, rather than the short repeat seen in the most eukaryote. So dekat fruit flies ataupun Drosophila melanogaster, so they have the transposition rather than the short repeat. Right? Dalam kebanyakan eukaryote. Okay. Nombor habis slide dah. Habis dah kan? Okay, habis. So, okay dah?
Doctor?
Sangat condense kan?
Doctor?
Yeah. Yes, Jordan.
I have a question.
Yes.
For the chromosome, like we have a telocentric chromosome, which it only have one arms, then how about its nomenclature? So there is no P or Q
Yes.
like that?
Yeah.
So
There's no arm.
Yeah, just the numbers.
Yes, just number.
Oh, okay. Thank you, doctor.
It depends. It's actually it depends also. If the centromere is on the top, yeah, so dia gunakan Q saja, dia tak ada P. Hello,
Yeah,
Jordan?
doctor.
Yeah.
So
If the there is that you just now you say about the telocentric kan? Right, what you
Yeah.
mentioned is telocentric. So if they are on the top, dekat sana, on the top, so P arm, they don't have the P arm, so we use Q to represent.
okay, now I get it, doctor. Thank you.
Okay. Depend on the positions of the telomere lah. Any more? Yeah. Okay, metaphase apa yang berlaku metaphase? Okay. Okay, you see, prophase, metaphase, anaphase, telophase, betul tak? So, at metaphase apa yang berlaku? What happened during metaphase? Are you sure? Are you sure? Metaphase. Metaphase dan light up kan? Light up equatorial kan? Ya. Itu masa dia belum pisah, betul tak? So to make sure dia nanti dia pisah, dia kena apa? Kalau dia nak pisah lah, kalau dia pisah tu dia nak to make sure that dia kena condense kan? Nah. Oh. Memang ada sebab dia. You kena tengok pada fasa keberapa. Okay? You kena pandai link tau dengan the cell cycle. Okay? Any more? Wah, 4:15 already. Okay? Kalau tak ada, then kita ada assignment, satu lagi assignment kan? Sekejap lah. Let me check. Okay, this is assignment 2. Yeah, tadi discussed in detail the principle and finding of these 3 early experiments in genetics and the Griffith, the Avery transformations, and Hershey chairs kan. So deadline is 5th of April. Okay, this is the second assignment, and the third assignment is create a drawing that focuses on the relationship between DNA, gene, chromosome, cell, and body. Then explain in detail the relationship between DNA, gene, chromosome, cell, and body. So there are 2 parts. One, you need to draw, and from the drawing, from the figures, then you explain. Tak perlu panjang sangat. I want to see your understanding. Boleh tak?
Okay, doctor.
Okay. All this assignment is helping you to score point. Because in exam, because it's MCQ. MCQ, I cannot help much. If you're wrong, you're wrong. Well, system will mark you wrong. But for assignment ke ataupun quizzes, assignment lah yang banyak saya boleh tolong. And then it's a best method for me to assess you also. I force you to read. And also it's more subjective. Saya boleh bagi markah kesian. Tapi kalau quizzes kah, ataupun midterm exam kah, ataupun final exam, tak ada markah kesian because the computer